Thank you, Eric, for that introduction, and thank you for inviting me. This is a really great event, and I can see how this is really fun for everybody in the Marshall community. I've had a great day, and as a matter of fact, the story Eric told earlier today, West Virginia Public Television asked me, what is the most amazing thing that you've ever seen in biology? And I contemplate telling them about the leech and the anus, but you won't be hearing about that on West Virginia Public TV. What I want to do tonight is talk about the tree of life, and it's an amazing amount of diversity that we see around us, and an even more amazing amount of diversity that we don't see around us. And what I want to try to do is give you an idea of this diversity of life on this planet through three examples. Here we go. We're going to climb around the tree of life. And the three major groups of organisms that live on this planet, over here, these are bacteria. These are archaea. These are single-celled organisms also. And this is Elvis, the common ancestor of all eukaryote life on the planet. What I want to do first is tell you the stories that I want to talk about. Again, I want to talk about, first of all, is there really a tree of life? Is there really a way to represent all animals, plants, bacteria that are alive on this planet in a bifurcating vertical tree of life? I want to also talk about the mother of all metazoans. We are metazoans. We are multiple-celled animals. That's what a metazoan is. And there are some really wonderful creatures out there that are also metazoans that we're searching for as the mother of all metazoans, as the prototype of all metazoans. And then, to not leave the green stuff out, I want to talk about Darwin's Abominable Mystery, which is a really nice mystery story. And I contemplated talking about DNA barcoding, which is at the tips of the tree of life, but we won't do that tonight. The tree of life is a NSF-funded endeavor that's been going on for the last decade. And as it says here, it tries to harness life's history to benefit science and society, and that's a pretty lofty kind of a goal for the tree of life. However, it does work. And what I want to show you tonight are some of the examples of how the tree of life is constructed, but also some of the examples of how the tree of life can be used to understand basic organismal properties. And those of you who read the New York Times or the Washington Post are familiar with these kinds of trees. Trees are all over the place. And before I go into these three stories, I wanted to give you some sort of a background for trees. Trees are used in all aspects of our modern life. This is a tree of languages, of human languages, drawn literally on a tree, showing the relationships of the various languages of humans. And this slide shows the popularity of trees as tattoos. This slide tells me that our graduate students have much more time on their hands than they really should have. These tattoos are, as you'll see here in a second, representations of one of the first diagrams of a branching tree to represent the relationships of organisms, in fact, from Darwin's notebooks and his 1838 notebooks. This is a tree from the movie School of Rock. This is Jack Black's tree describing the relationship of rock music. And you'll see right here, if you can't see it, blues, country, and jazz are the ancestors of all rock and roll. And as you see it fall out here, you end up with things like heavy metal and punk and things like that. So trees are actually used in a lot of different areas. Oh yes, and this one, this is a tree from South Park. This is Mrs. Gerritsen. This is after she turned in, Mr. Gerritsen, turned into Mrs. Gerritsen and had the affair with Richard Dawkins. And this tree is a confabulation by Mrs. Gerritsen. She didn't really want to teach evolutionary biology in her class, so she made this tree up, and you can see there's lots of really fairly offensive things in here. There are things that Sarah Palin would say we shouldn't say. So how do we represent the tree of life? How do we represent life on this earth? And we have to go way back in time to Aristotle. Of course, everything goes back to Aristotle. Everything we know goes back to Aristotle. And Aristotle came up with this great idea called the scala naturae. It was a great idea because it was innovative and it organized things. It said that we go from lower organized things like inanimate matter, like dirt, to lower plants, higher plants, et cetera, on up to man up here. And this way of organizing things is really interesting, but not so accurate. Oftentimes, we can get the scale wrong. If you don't get that one, then maybe you'll get this one. This scala naturae is really an excuse or an apology for the variation that we see on this planet. And it's a stab in the dark at how to organize life on this planet. And as we'll see here in a second, it's not a very good way to do things because it doesn't allow us to test hypotheses and it doesn't allow us to think about the nuances of evolution. And actually, the problem is it doesn't allow us to think about ancestors in evolution. So in order to understand trees and to understand the importance of trees, we need to go back to Darwin too. Now, we always, Aristotle, then Darwin. Usually people say, go back to the Bible or Shakespeare. For me, it's go back to Aristotle and Darwin. And here, what we see is what did Darwin do right? Now, this is a very, very, very smart man. If this man was alive today, he would be curing cancer, or he would be finding some unknown planet in the universe. This is the smartest man who's lived in the last 2 centuries, even smarter than Einstein, in my opinion. And what did he do right? Well, he provided overwhelming evidence for the occurrence of evolution. This had never been done before. And in his On the Origin of Species that he published in 1859, it was just one argument after another, after another, after another, after another. And if you read it, it's just wonderful. It's not boring at all. It's an amazing account of the existence of evolution on this planet. The other thing that he did right that no one else had done before is he provided a mechanism for how evolution worked, and that's natural selection. And natural selection was unheard of until Darwin came up with it, and also Alfred Russel Wallace, who simultaneously came up with the idea. Those are 2 things that everybody knows that he did, that Darwin did. But what else did he do? Well, he looked at variation, and he looked at it in excruciating detail. This is a passage from On the Origin right here, and it's about pigeons. Now, if you're from New York City, you don't want to hear anything about pigeons. But this is just an amazing description of all of the strains, all of the sports of pigeons. And Darwin had this amazing hold on variation. And variation caused him to change the way we think about the natural world. Up until Darwin's time, everybody thought in the context of types. You saw flowers out in the field and you would go, oh, those all look like a yellow flower. And that was it. You found the average. And what Darwin did was he said, wait a minute, the variation is really important. And that's what led him to the idea of natural selection. So he changed the way we think by making us think about variation. So he changed us to think about populations and population thinking. But the other thing that he did that is equally important, and he actually mentioned that it was as equally important as understanding natural selection, was he made us think in the context of trees. And this is essentially the lead-in to what I want to talk about with respect to these 3 examples. So in this quote from the Origin of Species, he says, "For on the principle of inheritance, all the forms descended, for instance, from A, would have something in common ancestry." Such a beautiful idea. And no one had done this before. In a tree, we can distinguish this or that branch, though at the actual fork, the 2 unite and blend together. So he's actually describing descent with modification and common ancestry here. And this is what trees are based on. This is what evolutionary trees are based on. And then he says, this is what we should be driven to if we were ever to succeed in collecting all the forms in any one class which have lived throughout all time and space. He described the tree of life right there. That's the tree of life. It's amazing. In 1859, this guy who had taken a voyage around the world on the Beagle, decided that the best way to represent diversity on his planet was through a tree of life. It's amazing. And not only did he do that, if that wasn't enough, he came up with natural selection. Just to drive home the importance of this with respect to Darwin, he also said in a letter to J.D. Hooker that he wrote just before, about a year before he published On the Origin, I will try to leave out all allusion to genera coming in and out, which doesn't make much sense to me or to anybody here, till when I discuss the principle of divergence, which is the tree of life, or which is this principle of tree thinking. And he says, which along with natural selection is the keystone of my book. And I have very great confidence it is sound. I wish I could write that in one of my papers. I wish I could just say, I have very much confidence that this is sound. It doesn't happen in modern science. And as a matter of fact, to drive this completely home, the only figure in On the Origin of Species, perhaps one of the most important books published in the last 300 years or 200 years, was a tree, this tree right here. It's the only figure that appeared in the first edition of On the Origin of Species, and it's a beautiful diagram of branching, of detailing of ancestors. You probably can't see it very well, but detailing of ancestors and descendants, etc. And this is the essence of what Darwin gave to us in the 1800s. Not only that, but natural selection. And it really comes from his thinking early on. This is from his notebooks, and this is the branching diagram that he drew in his notebook. And in one of the most beautifully understated ways of saying this is important, he wrote down, I think. And he thinks that this is the way that life exists on this planet. This is the tree of life. The principle of divergence that he was so involved in and so happy about. And in fact, if you look at this diagram, you can actually see what he's talking about with respect to trees and the principle of divergence. These at the tip, A, D, B, C, and if you look at those tattoos again, get online, look at those tattoos, those A, C, B, Ds are there too. What he's talking about is this right here is a common ancestor of all these right out here. And this thing right here, he says, must have had something in common with all of its descendants. And that's really neat. That's the major advance that he made in getting us to think about trees. So Darwin not only presented overwhelming evidence for the occurrence of evolution and a mechanism for evolution, but he also changed the way we think about the world, changed the way that natural historians think about what they see in nature. And he changed us to think about populations and variation. We would never have come up with natural selection without doing that. And he made us think about ancestors and descendants and the process of descendancy and how that relates to an evolutionary tree. Now, I wanted to go in just a little bit more detail before we get into the nitty-gritty of these trees. And when I get into the nitty-gritty of these trees, you might want to take a little nap because some of it's fairly technical. I think you might get the point. This should be familiar to most of you who've read Aristotle. Again, this is the scala naturae, and the scala naturae just says you go from earth to plants to animals to man. And this is, again, a fallacious way to think about the natural world and to think about how organisms have diverged from one another. But if you start to think about it in the context of what the ancestors of these things are doing, everything other than earth has what Aristotle called a vegetal soul. And everything except for plants and earth have what Aristotle called an animal soul. And everything except for animals, plants, and dirt have a rational soul, so to speak. Sometimes I question that. But the point is, if you think about things in the context, in the way that Aristotle thought, you miss all of this. You miss all of the ideas about what these ancestors are. So the ancestor of plants, animals, and man had a vegetal soul. And the ancestors of animals and man had an animal soul, as termed by Aristotle. And so on. If you go to the American Museum of Natural History, you'll notice that I was talking about this a little bit earlier, that the fourth floors in the museum are the dinosaur halls. And I have what's called Dino Envy because I hate dinosaurs. And that's a pretty strange thing for someone from a natural history museum to say. But I do hate them. And as I said to someone earlier this evening, when I started at the museum, I vowed that there would be another big D in the museum, and that would be DNA instead of dinosaurs. And I've failed miserably because people just love dinosaurs. But the fourth floor in the museum is actually an evolutionary tree. I don't know how many people have been to the American Museum of Natural History in New York City. Lots of you, yeah? And if you've been to the fourth floor of the museum recently, it was renovated about 10 years ago, you will have noticed that this is an evolutionary tree. So these halls, these hold the hall of fossil mammals. This holds the hall of advanced mammals. This holds the hall of primitive mammals. This holds ornithischian dinosaurs. This holds Saurischian dinosaurs. And this holds a bunch of things without legs. And so when you enter the hall, everything on the hall has a vertebrae. The ancestor of everything on that hall has a vertebrae. And then when you leave this part of the hall, everything in the rest of the hall has 4 limbs. The rest of the floor has 4 limbs. And then when you get out of this part of the hall and you're into here, everything that's in the rest of the hall has watertight eggs. And then when you get over here to the 2 great halls of dinosaurs, everything has a hole in the hip socket. That's the characteristic that defines dinosaurs. So what this does is it allows us to reconstruct the ancestors of organisms. And by reconstructing the ancestors of organisms, we learn more about how those organisms came to be, because those ancestors have to be some sort of a transitional form between the origin of life and those forms that exist at the tips of the tree. Now, this doesn't mean that fossils are ancestors. It doesn't mean that we can find missing links. It doesn't mean that Neanderthals live amongst us or anything like that. It just means that we can reconstruct the events or the characteristics that exist in life on this planet. And not only can we reconstruct the appearances or the anatomical characteristics that exist in animals, we can also use their genomes and reconstruct aspects of their genomes and understand how their genomes have evolved and made the transition from the ancestor of all life on this planet to the forms of life that we see today. Okay, now there are 2 simple kinds of questions that we can ask when we talk about the tree of life. The first thing we can ask is, what are the relationships of organisms? In other words, we look at the tree and we go, well, that's really neat because this thing's related to this thing and that thing's related to that thing. And oftentimes, that is enough. That's enough to excite a scientist into ecstasy and to give them enough courage to submit a paper to Nature or Science. The other thing you can do is, as I said, use the ancestral reconstructions or use the shape of the tree of life to understand how the natural world happened. You know, what are the events that produced all this diversity around us? It's pretty amazing if you look around us. There's plants, there's animals, there's humans, there's bacteria crawling around the carpet, there's all kinds of stuff. And to understand how that happened and how those different forms evolved, this is the other major thing that we can do at the Tree of Life. And so the first thing I want to do in climbing into the Tree of Life is actually climb at the root of the Tree of Life. Just start climbing up the trunk and ask, is there really a Tree of Life? And this is an important question because some scientists believe there is no Tree of Life. Some scientists believe that it's such a jumbled mess that we'll never extract it. And I want to make a case for there being a Tree of Life. This is the view of life before DNA sequencing. This is showing a group of bacteria right here, a group of eukarya, and a group of archaea. And there are 2 events that have happened here. One is cyanobacteria giving rise, jumping into plants, chloroplasts. And the other is this proteobacteria giving rise to something in all of eukarya, that's the mitochondria. So it was thought that there were 2 major events that disrupt the vertical history of life, and that is the transfer of these 2 bacterial genomes into eukaryotes to produce chloroplasts and mitochondria. But this is what we're seeing as a result of doing a lot of genome sequencing. And you'll see that it's a mess. I mean, I couldn't make any sense out of this figure, and I don't know how the author who drew it, but it implies that there's a lot of horizontality, a lot of horizontal movement of things that disrupt the vertical history of life. And in fact, some scientists have suggested that whole genome prokaryotic and tree of life phylogenetics has been viewed as a useless and inscrutable endeavor. Wow. That means that what I do for a living is useless and inscrutable. And it is safer to assume a comb-like topology of life, and the safe topology would be a soft polytomy, which in the terminology of phylogenesis is a comb that is there because we just don't know enough about those relationships. And so this is a really daunting question. This is a major, major question, and it's a major, major statement that the scientist who made this at first made. This is outrageous to a person who reads Darwin and who understands Darwin's principle of divergence. So the way we approached this is we took many, many whole genomes, bacterial whole genomes, archaeal whole genomes, and eukaryotic whole genomes. We then identified all of the gene families with more than one gain of a gene, because you can actually do that after you see the trees, and I'll show you this in a second. And then classify all of the changes as either being a gain or a loss. And if it's a gain, that means that it's a horizontal transfer event. Okay, that means that it's actually one of these things right here, one of these bad things that are happening in the background of the tree that disrupt the verticality of the tree. So then we test if taking those genes that we suspect of being horizontally transferred out of the data set affect the hypothesis. Okay? So this is what we're talking about. This is an evolutionary tree, and I'm going to show a few trees, not too many, but a few. But what this is it's a bunch of species up here at the tips of the tree, and this is just the shape of the tree. And each tree I'm going to show you here, the next 2 or 3 trees, are all the same shape. And if it's colored blue, it means it has a particular gene from its genome. And if it's colored yellow, it means it lacks that gene. And we can do this with whole genomes. We can actually ask, does this genome have this gene? Yes, no, and then go on with it. So in this case, this is a horizontal gene transfer because the ancestor, as we reconstruct it, has it. It gets lost and then regained here. And the implication is that it came from here over to here. That's a horizontal gene transfer. And down here, it's a little bit more complicated, just to show you that we have an algorithm that does this. But in this case, everything has it up to here, but then it's lost here and then regained here. And that's a horizontal transfer event. Okay? So what we do is we go through all of the genes and all of the genomes of these organisms and classify whether they're horizontally transferred or whether they're not. And the next thing we do, this is actually showing you that you can actually classify something as not horizontally transferred, even though it's got a bizarre pattern. In this case, everybody's got it, then it's lost, and it's lost. No horizontal transfer. Okay? It's okay to lose things, but to gain it, it's a little bit more difficult. So here's what we did. We took all of these genes and we took out we counted the number of horizontally transferred genes. And the idea that the guys who don't want a tree of life, who think a tree of life is a boondoggle, they think that the horizontal gene transfer has totally obscured the signal because there's so much of it. Okay? And what we're trying to show here is that there's not so much of it. These are the number of horizontally transferred genes. There are 1,000 of them. When you look at this big of a data set, there are close to 30 or 40,000 genes that are part of the data set, even though each bacterial organism has only about 2,000 or 3,000. And when we look at the genes that are not horizontally transferred, it looks like this. And just look at this line up and down here. This is a criterion that we use, that most people use to classify genes. And you'll see that there are 1,000 or so genes that are horizontally transferred, and there are about 6,000 or so genes that aren't. So there's a 6-to-1 no to yes horizontal gene transfer. This suggests to me that the rumors or the fear of horizontal gene transfer is not as bad as we think it is. The other thing that we can look at is that we can take those horizontally transferred genes out of the data set and draw a tree. And when we do that, what happens is when we keep them in, we get this tree, and we take them out, we get this tree. And this is bad, because when we take them out, we lose the resolution. We want more of this branching in the trees that we generate. And when we take the horizontally transferred genes out, we lose all sense of relationship. And this tells me that there is information in those horizontally transferred genes. It may not be good information, but there's information there that we need to deal with. And simply taking the horizontally transferred genes out is not such a good idea. So we came down to asking, is there really a tree of life? And actually, it's pretty simple if you think about it. There are 3 tests. If you put all the data together, that's what massively concatenated means. If you put all the data together into a matrix, does it give a resolved tree? If it didn't, then the game's over, right? If the tree is totally unresolved, like a bush instead of a tree, then no, there's no tree of life. The second question is, if you get a tree, is it robust? Is there statistical support for that tree? And if there isn't, game's over. There's no tree of life. And the final one is the resulting tree biologically meaningful? Meaning, do all archaea come out in one branch? Do all eukarya come out in one branch? Or do all bacteria come out in one branch? And if there's mixing, then there's no tree of life. It doesn't make any sense to us. So when we do this, we build a big matrix, 7 million characters. It's one of the biggest phylogenetic matrices constructed to date. And we get this tree, 166 genomes, whole genomes, from these are archaea here, these are eukarya here, and the rest are bacteria. And the tree is resolved. So test number 1 wins. I just blown this up to show you that a D here means that it's supported with a probability of 1. So the data are saying that all of the nodes in this part of the tree are supported with a probability of 1, meaning that, yes, it's robust. And then finally, the relationships that we see here actually make sense. All of the eukarya are in a group. All of the archaea are in a group, and all the bacteria are in a single group. As a matter of fact, all the major groups of bacteria are in a proper group, and all the major groups of eukaryotes actually conform to what we think eukaryote evolution has occurred. And so these 3 tests for a vertical tree of life actually get passed. And what this tells me is that there is a vertical tree of life. This is very important, because if there wasn't, then we would have to deal with all of this mixing and matching of genomes that has occurred, or that people think has occurred, between eukaryotes, archaea, and bacteria. Okay, let's climb a little bit further up the tree of life and go into the branch that is called metazoans. And in this case, we're going to look at what the mother of all metazoans is and why this is important. And the first thing that I've told you about is more about the shape of the tree of life. And the next 2 things I'm going to talk about is how you can use the shape of the tree to actually say something about function and structure of organisms. So you have probably all learned this in your basic biology course. You have sponges out here as coming into the common ancestor of all metazoans. So this would be called, improperly would be called, the most primitive metazoan. Then you have this thing called placozoa. Placozoa is a beautiful animal. It's 3 layers of cells, and it moves around. It has what people call a top and a bottom, but the top and the bottom have no resemblance to anterior and posterior. So it's a really beautiful little animal. It's got a 4th cell type, and that's its gonads. So this is streamlined. This is streamlining, baby. You know, you got three layers of cells, and you got your gonads, and that's all you need. So it's a beautiful animal. And then you come into this group right here, where cnidaria, things like corals and medusa and jellyfish and things like that, and ctenophores, the comb jellies. The comb jellies are just beautiful. You can look at them online. They're metazoans that have just amazing structures. They're beautiful. And then this is us, Bilateria, represented by this Nemo fish. This is the classic way of thinking about how metazoans are related to each other. But if you look at the literature, every possible arrangement of sponges, cnidaria, ctenophores, Bilateria, and Placozoa posed, every possible one, and have been supported by different studies. That's just what this slide shows. You don't have to look at it very closely. So in my lab at the museum and in collaboration with a lab in Germany, we constructed a huge matrix of nuclear and mitochondrial genes and made a tree based on those sequences. And here you see the hypothesis shift. And this is a typical thing that happens when you do phylogenetic analysis. A hypothesis is always a moving target. Let me put it this way, reality is always a moving target. And this is the beautiful thing about science, and that makes systematics really kind of beautiful, is that you're always chasing this elusive truth with your hypotheses. And so what we see here is Monosiga, which is a choanoflagellate. I didn't point that out in the previous slide. It's a single-celled organism that lives in communities that does some really bizarre things. Suffice it to say that that's what it is. And then you have Placozoa. Now, in the other tree, sponges were first, right? So now we have Placozoa coming off first, and then we have sponges. Then we have Cnidaria and ctenophores, the comb jellies and the jellyfish. And now the Bilateria are the last thing off. Actually, they're the first thing off. So the Bilateria are actually the mothers of all metazoans. And in this tree, Porifera are the mother of all metazoans. So our group, Bilateria, in this tree, the tree that we generated at the museum and in Germany, it says that the Bilateria are the mother of all organisms. Then another study came out, and it said that the ctenophores were the mothers of all organisms. So what I want to do now is actually show you why some people think one hypothesis is better than the other. These are all equally supported from different sources of data. These data are completely different from these data, are completely different from these data. So if we think of these organisms in the context of a nervous system, because animals are unique with respect to having a nervous system, we can see why people prefer this. And that's because you only have to hypothesize the evolution of a nervous system once. So here we have Cnidaria, ctenophore, and Bilateria, which all have so-called nervous systems. Cnidaria and ctenophores have what are called neural nets. These are not a classical nervous system, and nor do they have a brain, though. They have a neural net. But most Bilateria have either a neural net, like in echinoderms, or a nervous system and a brain. And so you only have to hypothesize the origin of a nervous system once in this hypothesis. Over here, in this hypothesis where ctenophores are the mother of all metazoans, you have to hypothesize two events. You get it, and then you lose it. And if you notice that Placozoa aren't in here. If you throw Placozoa into this data set, you actually have to hypothesize three events. So ctenophores as the mother of all metazoans is not a very good hypothesis to actually sit on. Over here is the one that we generated. It hypothesizes that you have to get two gains of a nervous system. So this is why people, when they saw this publication, said, oh, it can't be, because nervous systems only evolved once. But when you start to look at the genomes of these organisms, you start to see some really interesting things. And here we see fungi, which are the closest thing to metazoans out there. In fact, fungi are more closely related to us than either we or fungi are related to plants. That's how closely related they are to us. So we have fungi, choanoflagellates, that funny single-celled organism that forms a colony, poriferans, cnidarians, protostomes and deuterostomes. These are both bilaterians. They're us, really. And this is just a list of genes that are involved in synapses. Synapses are the junctions of neural cells that make up our nervous system. And you can see that Porifera has about half the genes to make up a synapse, which is pretty amazing in and of itself, because sponges don't have synapses at all. They don't have anything that at all resembles a nervous system. And Cnidaria that have the neural net don't have quite as many of these genes as protostomes or deuterostomes have. So the idea here is, well, this kind of shores up the idea that cnidarians have a protosynapse, and protostomes and deuterostomes have real synapses, and sponges don't. But if you look at if you throw in a much more detailed way of looking at these organisms and actually throw the Placozoa in, you find that you will find these genes in sponges if you do a much more aggressive search of their genomes. And you find that Placozoa actually has all of these genes too. Now remember, Placozoa has 3 cell layers, right? And it's got those gonads hanging around too. But it's got these 3 cell layers and no semblance of a nervous system whatsoever, no synapses. It's a very, very simple animal. Yet it has all the genes for a nervous system, which is really bizarre. And this tells me that you have to look at the nervous system of these organisms in a completely different light. All the genes are there. And it tells us that there's something really interesting going on here. And so if you actually separate the nervous system into whether or not these things have genes, the genes for a nervous system, whether or not they have a stimulus perception, whether or not they have stimulus conduction, whether they have functional nerve cells or specialized nerve cells, the story becomes a little bit different. Instead of the hypothesis with sponges as the mother of all metazoans being twice as good or 100% better than the other 2, now it's only about 20% better, because you can map these characters onto the tree that we generated with 6 steps, 4 here and 1 there. So the specialized nerve cells arise in bilateria and cnidaria and ctenophores. And over here you have, this is wrong, it's 6 or 7 steps. And here you have 5 steps. So instead of being, like I say, 100% better, instead of this hypothesis being 100% better than, say, this one, and 200% better than this one, it's now only about 20% better. It's still better, and I'll give you that, but it is not such an open and shut case now when you start to dissect the nervous system. Okay, so we're almost to the very end of climbing up this tree of life. And what we're doing now is we're climbing back down out of metazoa, past fungi, and up plants, okay, because plants are outside of fungi and metazoa. And we're in a situation where we have what we call Darwin's Abominable Mystery. And Darwin's Abominable Mystery refers to the fact that the diversification of flowering plants from primitive gymnosperms, which are oftentimes left out of plant phylogenies, as the abominable mystery due to their sudden appearance in the fossil record, which is really bizarre because almost all of these plant forms right here occurred, bang, all at once. Really, really interesting. And Darwin knew this. He knew these were fossils, and he knew that this had happened. And he was just, like any good scientist, he's scratching his head saying, how did this happen? This is just really, really amazing. And you have to go back and think about his ideas about the tree of life and descent with modification. Remember, he said that the descendants should have something in common with the ancestors. But these things, if you look at them and try to decide what is in common in the ancestor, it's really hard. So this became what he called his abominable mystery. He called it this in a letter, once again, to J.D. Hooker, his good friend in London. And so what we've done at the American Museum and at the Cold Spring Harbor Labs and NYU and the New York Botanical Garden is start to look at gymnosperms in order to understand how the abominable mystery started. And these stars here indicate large-scale sequencing projects that have been done on several taxa in this outgroup Selaginella, in cycads, ginkgos, and in Nitales, which are really interesting plants called gymnosperms. Now, gymnosperms don't flower. They're the non-flowering seed plants. Everything above here, not ferns and not Selaginella, are seed plants. And these are non-flowering seed plants, and these are flowering plants. And so Darwin's mystery actually involves this over here. But in order to understand that mystery, in order to understand how to solve that mystery, we need to know what was happening in these gymnosperms. And once again, just like the metazoan tree, many, many hypotheses have been offered as to the relationships of Nitales, angiosperms, ginkgos, cycads, and conifers. And there are at least 4 major hypotheses that people argue about, and they're described over here in more detail. So what we do to do this is we created a huge matrix. The the theme of the evening is huge matrix. And the matrix consists of DNA sequences and protein sequences from about 30,000 genes in over 160 plants. So this is a study that was completed about 3 months ago, and there are already double that number of plants in the database. So we were a little afraid of that. We were afraid of writing the same paper over and over, so we automated it, and we now have a website called Big Plant that automatically updates itself and builds a matrix and builds a tree. It's truly a growing tree. It grows every time a new data set is deposited into the database, and it grows by adding one more branch to the tree, which represents the new taxon that's added to the database. This is perhaps the only way to do this because technology is just moving so fast. So you can go to this website, Ortholog ID Big Plant, and take a look at it and download the matrix and play with it yourself. This is the tree. It shouldn't mean that much to you, other than to look over here and notice that gymnosperms all have a common ancestor. Angiosperms all have a common ancestor right here. Actually, it's right here. Monocots, which are things like grasses and corn and things like that, have a common ancestor. And the dicots right here all have a common ancestor, which is good. This is really great. And in fact, the problem we have in trying to publish this is that it's nothing new. This is the comment that we get from reviewers when we try to push this through review. And this is a complete misunderstanding of how systematics works and how science works. This is the largest phylogenetic data set for these organisms that's in existence, and it's corroborative. It corroborates the hypotheses that people who use chloroplast DNA are specific to plants, to construct trees. That's, in and of itself, it's important to corroborate the known hypotheses with as much data as you possibly can. That's how science works. The red dots indicate the strength of the inference. Anything that's red, 1.0 probability. So we can use this tree, though. The shape of the tree in and of itself is really important in corroborating other hypotheses. But it's also important because we can now use the tree to understand Darwin's abominable mystery. And what we can do, what people have done in the past, is use trees to search for natural selection. They actually have used the relationships of organisms to search for the existence of natural selection. And anything that's naturally selected, that is under natural selection, any gene that's under natural selection, is important for that part of the tree. And the other way is to trace characters on the tree, like I did with the presence-absence of genes in the bacteria and the archaea and eukarya. So this is a really interesting study that shows how you look for natural selection in genomes. These are all domestic chickens. This is the red jungle fowl right up here as kind of an ancestor of everything. And researchers have sequenced the genomes of all of these guys. This is a white leghorn, obese strain, Rhode Island red, low growth line, high growth line, broiler, two broiler lines. And they've sequenced the genomes here. And over here, what they've done is they've estimated the amount of natural selection that occurs in a particular chromosomal region. And the colors represent the 21 chromosomes. I think it's 21 chromosomes that chickens have. And you can see there's a line drawn here where on this side there's no selection, and on this side there is selection. So what they've done is they scanned through the entire genome for all domestic lines. That would be all of these guys down here, to look for natural selection. And they can identify all of these genes here as being important in the selection of these lines for domestication. And if you look at commercial broilers, that's these guys right here. You see a different profile, fewer genes. And when you look at the layers, that's these guys down here, the obese strain, the white leghorn and the A and the white leghorn B, you see even fewer genes that are under natural selection. There's a phylogenetic effect here, and what it's done is node by node. You're actually looking at this node and asking what's under natural selection, looking at this node and asking what's under natural selection, and this node, what's under natural selection. Now another example is this, in yeasts and fungi. These are actually bilateria here, but these are all yeast here, fungi and yeast. And these are 4 genes that were mapped onto the tree. And so what's interesting here is that when you do this gene by gene, you end up with look here, these two genes here arose in the common ancestor of Candida albicans. These are all yeasts. And so these two genes can therefore be thought to be important in the evolution of yeasts. And just as a contrast, you can see over here where there is a presence and absence over here, there's no correlation of presence or absence. Actually, no correlation of presence or absence of those two genes. So if you do that gene by gene, you can end up with genes that are thought to be important in the evolution of the traits that are important in these yeasts and fungi. Well, the real way to do this is node by node plus gene by gene, to put them both together and to actually look at each gene at each node. And this is a huge computational problem. It's a huge bioinformatic problem. And even if you do that, not all evolution occurs as a result of natural selection. So you don't want to look for natural selection either. And in fact, this is a diagram that just shows what happens if natural selection is at work. This many genes, or this proportion of genes, are under natural selection and would be detected by these selection tests that I showed you for the chickens. But if most of the genes in the genome are evolving neutrally, then only this proportion of genes will be detected. So we need another method, and if you use what's called a nearly neutral theory, then this proportion of genes would be detected. A very, very small proportion of the genome gets detected. So what you need to do is actually look for change. Anything that changes at a node for a particular gene now becomes important for that particular node. Rather than relying on natural selection, why not just look at change? So a gene can either agree with a particular node in a tree, or it can contradict the node. This is what I mean by agree or disagree. So we see the same tree that we drew for plants. These are gymnosperms down here, and these are all the angiosperms up here. And what we see here is a bunch of genes across here. These are all different genes, and the gene goes down the column here. And wherever you see something orange, that means that that gene is supporting that node. So for instance, if we look at the node for all angiosperms right here, we see that there are about 15 genes that have strong support for that particular node. This means that these genes are actually involved in some aspect of the evolution of angiosperms. And also up here for asterids, we see that there are about 20 genes that are involved in the divergence of asterids. So what we're able to do here by using the whole genome sequences of these plants to focus on the genes that are important for their divergence, important for their diversification. And in essence, by looking at this particular node right here, we can try to solve Darwin's abominable mystery. Why did all of these angiosperms just burst into diversity? And when we look at this, we can look at up here these are the rosids. We see that we have radical detoxification in species, well-known sources of antioxidants. These are genes that are important in antioxidants that are important in rosids, in the rosid families. If we look at the next thing, the caryophyllates are down here, and they're a strange group of plants because they use sulfur compounds. And in essence, what we discovered when we did this analysis is that sulfur compound metabolism and drought stress is really important at this node. These are the genes that we extract using this procedure. And down here, to kind of address Darwin's abominable mystery in the monocot clade, we see that cell fate commitment and RNAi and tacrRNA, which are small interfering RNAs that work in gene regulation in plants, are responsible or at least active at that node and point to those particular genes as a possible way of thinking about how plants diversify. These are the genes that we can look at now and start to do experiments with to see if they are actually involved in Darwin's abominable mystery. The problem that we have with a lot of this is that there are so many genes in a genome. It's to do 30,000 transfection experiments is really quite hard, almost impossible. And so what we need to do is winnow the possibilities down, and this approach using a phylogenetic tree and using the mapping of characters and the description of how these genes are supporting particular families of plants or groups of plants allows us to narrow the possibilities down. It's not much of a leap to understand how we might use, say, a tree of papillomaviruses, human papillomaviruses, or how we might use a tree of Staphylococcus strains, or how we might use a tree of Leishmaniasis strains, or a tree of Plasmodium strains, to try to understand the diversity of different kinds of infectious diseases. And it's not too far of a stretch for you to imagine applying this same technique to different mammal groups, or different plant groups, or different fungal groups, et cetera, to try to understand the diversity of life on this planet. And that's pretty much all I have to say, other than to acknowledge my collaborators and the mother of metazoans, Baron Shearwater, Sergius Colocotronis, and Mishi Eitel, a Tree of Life work with Paul Planette, who's a MD, PhD, a brilliant kid, and Kurt Leno, and the Abominable Mystery with Gloria Caruzzi, Angelica Cibrian, and Ernie Lee, and the New York Plant Genomics Consortium. And thanks for listening to me. I hope you got something out of trees. Thank you.